the computer. Look at that. Okay, so again, apologies for yesterday where I forgot to do that. And then I've got the PowerPoint somewhere in here, Blackboard. Still in Unit 3. Chapter 12. That's where we're going to focus. And can Zoom hear me? Okay, good deal. Share screen. That should be being shared now. Okay, now we're good to go. All right, so Zoom, if y'all have any questions, just chime in, break in. So molecular diagnostic techniques, and you go, well, that's just for big labs, right? And really not. I was working in basically a 10-bed hospital. I mean, we could put more in there than that, but that's what the DeWitt Hospital, basically, it would go 10 to 20, but we very rarely got to 20 patients in-house. But the lab all of a sudden came up with this idea that they had a salesman come by and they said, I have a BioFire, okay? A BioFire molecular analyzer, detect molecular, and you can do blood cultures with this, you can do respiratory with this, you can do stool with this. So don't, start putting your samples in here and you will identify within an hour what organisms are in that sample. So you're thinking, well, that sounds like molecular, and it was, okay? It was really basic, it was really easy. You had to keep everything, wore a mat, a face shield, gloved up, you sprayed down, and then you put your sample in, measured it, put it into a film array pouch, and it disappears into a little box, like a bread box, and it starts doing its thing. And then 55, 60 minutes later, it would say finished and printing off the result, and then it would give you a list of detected organisms, okay? So that's kind of where, saw that. It was like, oh, we'd always heard about different things, but now it had been brought into a small 10-bed hospital lab and being used. The doctors loved it. So the doctors would say, okay, I've got diarrhea, go down there and do a film array on it. I want to know, do I have something I need to worry about? You detect the stool organisms that we worked on in micro, right? Sorry, Preston's back there. Sorry about you hadn't worked on these, but we worked on those in micro. So instead of taking that, plating it on Hectoin, looking for Salmonella Shigella, we just take that stool sample, put it in a pouch. An hour later, we'd be able to tell the doctor if we had Salmonella Shigella, C. diff, anything that could be causing the diarrhea, did we have it there? And that's what they wanted. And that was a really great tool. I loved it because it was complete. I mean, it was quick, it was clean, and it gave results, okay? Now, some of the little hurdles that were problems were the patient have private insurance or they have Medicare. So we had to deal with that because some of this wasn't approved to be reimbursed that quickly or that much for that kind of test. So we were looking at, $300+ to charge the patient for that. So you can imagine if you got that bill and your insurance didn't cover it, you're stuck with a $300 bill for the doctor to run a film array on you. But it was that, and if you haven't been in the hospital setting, the administration part of hospitals like that because they like big dollar test reimbursement. They love CT scans from X-ray, and they love film array molecular testing from lab. Okay, they don't like the CBC. Y'all just started the CBC, right? Hematology. You know, that's a $2 test. That's administrators aren't really excited about a $2 reimbursement. But you give them something that's $350, $500, or $1,000, like CTs are, they're all thumbs up, and they want to make sure you're staffed to do that. And one thing one administrator once told me was, lab has to do a lot of tests to make up for like one X-ray test. Okay, so if he can get a CT done on a patient, that's like 20 lab patients, all right, to add up to get to that. So that's kind of where we are in healthcare today when we think about molecular. So we're going to go over DNA and RNA, and this is where I don't know how educated you are. I think you've had DNA and RNA since junior high, but do you really know what DNA and RNA look like, and the names and of the bases and the sugars? So we'll do a review of that. Then we're going to look how we do molecular analysis, and then DNA sequencing is where we may or may not get within this 50-minute lecture. So why do we want to do molecular diagnostic techniques? I hope you got some info from my intro there I gave. Used to gain information to aid in diagnosing, prognosing, and monitoring. So it's this, you know, do we want to wait on something, or can we know exactly? And your book chapter goes into just, you know, thinking about, you know, criminal cases. Just think of that with DNA testing, right? People are getting let out of jail because they can't prove by DNA testing that that person was actually at the crime scene 20 years ago, right? The technology has finally caught up to that case, and they saved all the stuff, right? Which makes it great for DNA testing. And now we can say, you know, there's no proof this person was even there. There's none of his DNA or her DNA at the crime scene that you collected 20 years ago. Thus, they appeal the case, and they usually get let out based on that, right? So think about that, how far we've come just in the last 20 years on DNA testing. We're going to detect specific nucleic acid sequences in microorganisms. Now, the good thing about microorganisms is their DNA or RNA is not that expansive. You know, it's very simple. So we can do things with microorganisms that you can't do real readily with human, okay, DNA sequences, because it's just more complex. And then we can take cells before antibody detection is possible. So, you know, we're doing with COVID, right? COVID-19. The reason we wanted to do PCR testing, the reason that was the ideal setup for testing, was we didn't know when the antibodies would come up, right? So you're waiting on an antibody response from a patient that may or may not show symptoms. So the PCR test was the original, like, let's get the PCR and detect the viral DNA. And that way we'll know if you actually have the virus infection. So this is definitely in the news today. So here's our DNA and RNA review. So you see purines and pyrimidines, and you need to know which is which. Can't just have you blunder this, right? Because we're going to talk about, in clinical chemistry, you talked about one of the, I'm going to test really. So I got one student here who's been in clinical chemistry. Huh? Tristan too? Okay, so between the two of you, what is gout? Y'all remember, did y'all talk about gout?
Like the disease?
Yeah, talking about uric acid, right?
Yeah.
Excess uric acid sets up in the joint, causes this painful joint, usually in the big toe. But when we detect it, what is actually causing the increase in uric acid? Uracil, right?
It's not up there.
It's not up there, right? Well, it could be, it will be here in a little bit, because it takes the place of who? The T gets replaced by uracil, right? that right? For RNA? Is that correct?
It's one of the two bottom ones.
One of the two bottom. Well, there's one that's common for, 3 of these are common for DNA and RNA, and there's one that's not common for RNA. Right.
Yeah, it's the T.
All right, thymine, okay. All right, so amino acid. Right, so we'll get back to that a little bit. DNA carries the primary genetic information within the chromosomes, right? And y'all know how many chromosomes we have? 46, 23 pairs, right? Okay, all right, good. That's good to know. You have purine bases, which are adenine and guanine. So those are our purines, A and G. And then they link up with pyrimidine. So in this base pair, you have one of each. So that should be something you know already too, but you may not. And we have it lined up pretty well in the picture that you know the A of the purines binds with the thymine of the pyrimidine, and the guanine binds with the cytosine, right? The base pair of the DNA. DNA is a double helix, comes across, and these base pairs are linked to make the ladder of the DNA. We all right with that? Everybody, that's basic. RNA is an intermediate nucleic acid structure, and it does help convert the genetic information encoded in the DNA into protein. So there's going to be this, DNA opening up. There's going to be this translation, then there's going to be a transcription, and then we're going to get base pairs, and then we're going to get a sequence of messenger RNA, and that's where the protein ribosomes bring in the amino acids as they read the sequence of the RNA, which is what? It is a mirror image translated of the DNA, right? Okay, we'll see that in just a minute. But we did get a change here with pyrimidine. We have a uracil. So this slide did not match them up very well because it didn't match up adenine and uracil, and guanine and cytosine are still matched up. Okay, and mainly because RNA is usually in a single strand. Okay, we don't see it in a double strand formation like DNA. So DNA and RNA both have nucleotides, and this is how we set these up. So you have a phosphate group and a deoxyribose here for this is DNA, right? So you have a carbohydrate, you have your phosphate, and then you have your amine of the base. So they're nitrogenous base. So you see a lot of nitrogen here and amine groups. And then over here you got your phosphate. Okay, so the nucleotide is the phosphate plus the carbohydrate plus the base. So the whole thing is known as a nucleotide. So the ribose sugar is either deoxyribose or just ribose for RNA. So this shows you the DNA double-stranded here with the A and T, adenine and tyrosine, or thymine. Which one is it? Tyrosine, thymine? Thymine? Thymine, okay. Guanine and cytosine together, okay. And you know that the DNA runs in an opposite direction. So it goes from 5' to 3' and 5' opposite. So the two rungs of the ladder, the two outside, not the rungs, but the two sides of the ladder run in the opposite direction. Okay. And then here's RNA, here's a single stranded DNA, and then the RNA shows you the uracil taking place of the thymine. Here's the DNA double helix. You see the nitrogen bases with the sugar phosphate groups. The sugar phosphate groups are out here. Okay. The nitrogen bases are inside, and you see the A and the T and the G and the C making up the rungs. These are the rungs of the ladder in the double helix of DNA. So how does DNA replicate? There are two strands of DNA will separate. Okay, so that double helix opens up. It doesn't like disintegrate, okay, because it's going to be like a zipper, it unzips itself. And we've got something that does that, and you're going to need DNA polymerase to do that, okay. Each strand is a template for synthesis of the complementary strand, and each new strand is synthesized in an opposite direction. So if you're going from, 5' to 3', you're going to go in the opposite direction, and we'll see that in just a minute. So you end up with two identical double-strand molecules known as daughters. They're at the end of DNA replication. I think I've got a picture here to show that. So the original strands are here on the outside. Okay, so this out here is the original strand. Oops, do that. Oops, there we go, back to that. And that's the zipper part. So if you look at, here is what the original strand, what we're looking at, you see it unzips. And then you got DNA polymerase, you got DNA ligase. Ligase is removing, breaking some segments. And you got RNA. RNA primase, and what that is that you have to have a little start. You have to have a little piece of RNA to get this DNA replication started. So that's all that RNA is in there for right now, because we don't think of RNA, because these are daughter strands of DNA. Okay, because if we're doing this inside the cell, then that's what's going to go to the new cell, or the daughter strands of DNA. Okay, so as it opens up, you're going to copy, if you want to think of it that way, it's a copy of the sequence of DNA. And you can see that if there was something that went wrong or amiss during this process, it could cause problems for the new cell. And think that down the line, like what happens during a mutation, what happens during a process that's not really good, if we have an error. So this does it millions of times over and everything's okay, but then sometimes we can have a hiccup, and you hope there is a mechanism to catch that hiccup. Okay, and I look at it as simply one of my best stories ever. You know that ultraviolet light is never good for us, right? So if we're out in the sun, we have damage to our skin cells, right? And that's usually corrected. There's a repair of our skin cells with the ultraviolet light is causing these problems or mutations or whatever. Think of it that way. So one of the things I always hint at is that's ongoing every time we're out in the sun. But one of the things that blocks that repair is caffeine. I always like to tell that story because if you're out in the sun, you don't need to be drinking caffeine. Okay, because that ongoing repair of your DNA that's happening in the skin cells is blocked by caffeine. So you always need to not drink caffeine when you're on the beach. Okay, if you're hanging out on the beach, or if you're hanging out outside at a football game, or if you're just hanging out at Craighead Forest walking trails, right? Your exposed skin doesn't need caffeine. It needs no caffeine to block repair of the damage UV light causes. And that's just natural sunlight.
That's difficult.
Hmm?
I said that's difficult.
That's difficult. I know it's difficult. It's very hard to think that way, but you got to try. Got to try.
You should replace it with alcohol.
Huh?
Yeah, you're right.
What's that?
I don't know.
I didn't hear you. Did you say, what did you say? Did you say something like, that's why you drink something besides caffeine when you're outside? That's what you said?
I said you replace it with alcohol.
That's what I thought you said. That's exactly right. So yes, you're doing wonders there. You're helping out your cells by staying off the Cokes and Dr. Peppers, and you're going with the other. Okay. So RNA synthesis, okay, Yeah, it can start de novo without the primer, because we know we had to have the primer of RNA to get that DNA replication started. So RNA can just start up and do its thing. It can be replicated or synthesized. It's catalyzed by RNA polymerase, which makes sense. We had DNA polymerase as being the reason DNA could do this. And it occurs throughout the life of the cell cycle. Now, a little flashback to biology, right? Y'all learned the cell cycle in biology. Y'all remember it? G1, S, and G2 phase. Remember that? As you went through mitosis.
That was like first year class.
Remember that?
Yeah.
Remember that way back when? Well, anyway, there's the S phase is when only DNA synthesis occurs, but RNA synthesis can occur throughout the growth, the cell cycle. Okay. Just seeing if y'all can remember back. So RNA codes for proteins, and we kind of know that. Well, hopefully we know that. We know that, you know, from the cell biology part of the nucleus, we know the DNA is in the nucleus, right? And we know that RNA comes out of the nucleus, right, through the nuclear pores, and then it sets up outside, and then that's where the transfer RNA and the codons of amino acids get read off the messenger RNA, and that's how the proteins get built. Y'all know that, right? I hope you know that. It's just basic stuff, but I don't know if you know it. That's why I'm asking it this way. Y'all do know that happens that way, right? I got so frustrated one time I bought a DVD for class. I got tired of just going over DNA, RNA so much that I said, well, maybe I need a video to show. And then I would just put it in and I'd leave and go to my office and say, okay, you guys watch RNA, DNA, and protein synthesis, right? Well, I don't want to do that. So I like this chapter. It's hard. It's harder than the others that we've read so far, but we need to kind of get a grasp of just basic molecular. Okay. Transfer RNA brings in the amino acids. Ribosomal RNA goes across the messenger RNA, bringing in amino acids to form the long protein. It's just a strand. So here it is. This is protein synthesis and translation. So here's your DNA, all right? DNA is inside the nucleus, all right? And there's transcription, okay, that changes DNA into RNA. So that RNA reads the pattern of DNA, right? Then you get that's transcribing, and then translation is the reading of the RNA or the messenger RNA that was produced after transcription. This is the messenger RNA down here. This is the ribosomes, right? What is it, 50S, 30S, 20S, 70S, 70S, I think. 70S, right? And that's just, that's the centrifugal weight. That's where that S comes from. And then you see this polypeptide chain of amino acids coming off the messenger RNA being read, okay? So you do have a list of amino acids in your textbook. 173 gives you the list and the genetic codes. Anybody had biochem yet? Okay, this is big in biochem, okay? Those codes there, all those amino acid codes, those start codons, those stop codons, right? Because that determines the amino acid sequence. So to build a protein, this is how we get it, right? We've got to have a certain amino acid come in where it's coded for. Okay, so this is like the building block. So you see the 20 amino acids: alanine, arginine, asparagine, aspartic acid, cytosine, glutamine, glutamic acid, glycine, histidine. And you see their codes, the codons over there in a 3-base sequence, GCU. What would GCU stand for? What would GCU stand for an RNA? Yeah, that's just those base pairs we started with, right? It's either G, C, A, or U for RNA. All right. So other DNA and RNA characteristics, we do get mutations, and we kind of hinted at those earlier. But a mutation is a change in the nucleotide sequence. And when we get a mutation, we can have cause immunodeficiencies or affect response to therapy. Your book talked about one, and I think I noted it because I was like, I need to talk about it because it had something to do with hematology. See if I can find it real quick. What I'm looking for is the sickle cell mutation. Anybody, did y'all talk about sickle cell? Y'all went through red blood cells already, right?
I think sickle cell is the next chapter that we're doing.
It's what?
I think it's the next one we're doing. Like on our syllabus, sickle cell.
Oh, okay. Y'all hadn't quite got there yet? Okay. I was thinking it was in here. I remember reading it. Oh, here it is. I found it. On page 174. A well-known example is the A to T base substitution, okay, in the beta globulin gene on chromosome 11. When that happens, it causes sickle cell anemia. Okay, so we can get a DNA substitution, results in the replacement of glutamic acid with valine at the position of 6 in the protein sequence. And what that does, it gives us an abnormal red blood cell that does not circulate efficiently. We know that about sickle cell, and the deleterious effect is balanced by a beneficial phenotype resistant. Okay, so you get a benefit if you're in a malarial infected plasmodium, and we'll do that one next semester in parasitology. But that's what it is, right? So this mutation actually was beneficial. I know we see that now as not beneficial because we're not in a malarial infected area, and we see patients that come in with sickle cell and they're in pain and they're having potential clots and they have painful circulation, right? And we have to treat them with fluids, but at one time it was survival. Okay, so that mutation that happened was a survival mechanism because it decreased the effects of plasmodium getting into the red blood cells. So you get infected by the mosquito, but it couldn't infect the red blood cells, so it ended there and you didn't suffer from malaria. All right, here is a word that we'll see a few times. It's restriction fragment length polymorphisms. Okay, so RFLPSs, this is on page 175. Polymorphisms that create, destroy, or otherwise affect restriction enzyme sites. Okay, so different variations that differ among individuals. They're caused by variations in nucleotides within genes that change where restriction enzymes cleave the DNA. So some of our testing that we're going to hit on first is just cleaving the DNA at a certain point. Find the sequence and cut it, right? We have enzymes that can cut DNA. But that is one of the words, the little abbreviations I wanted to make sure you had at least what it meant, maybe not what it does or what it results, but maybe just know this restriction fragment length polymorphisms. So we can get some polymorphisms or mutations that are defined by the frequency in a population. So if we checked all of us, right, that all of a sudden everybody had this mutation, right, common amongst a certain population, could be a certain race, could be a certain group, a certain nationality, right? We can find those. We can find those by doing testing. All right, so we're moving into the molecular analysis. So we'll kind of get started with it. And we'll get a few talked about today, and then we're going to break and then come back on Monday and finish up the chapter, because as you keep going, you get into DNA sequencing at the very end. We're going to do more. We're going to spend a lot a little more time on the molecular analysis. We got about, what, 15 minutes left, is that right?
Yeah.
And that's about right for today. So molecular analysis, we're going to start with this. We're going to start with the strand cleavage method. So first one up in green here on 175 is the strand cleavage method. So the nucleic acid test designed to detect changes in the DNA sequence, there are 4 approaches. There is the nucleic acid analysis. That's one. This is on 175 too. There is the strand cleavage method. There is the hybridization method, sequencing methods, and the amplification methods. So that's kind of, we got strand cleavage is the first one, hybridization method, sequencing, and amplification. So those are the 4 that we're going to work through first. We're going to try to work through these and what they're good for, how they actually go about doing their business, which one ends up being the most popular of all, because that's the one you'll see. Hopefully you'll get in a lab that does some of this. Some of the issues we've talked about before is they may have the technology, they may have the platform, but they don't have the supplies right now. So somebody might have PCR testing at the NEA clinic, the NEA hospital lab right now, but they can't get the supplies. So they don't have the test reagents. They don't have the test cartridges to run PCR testing on their swabs, okay? So that's been the biggest issue from March to now is that, yeah, we have these platforms, but you want us to do 10,000 tests a day, right? We don't have enough stuff. We don't have enough stuff to run that many tests a day. And that's kind of where we got overloaded as the lab. The lab kind of got looked at as maybe we didn't know what we were doing. Maybe we didn't know how to handle this kind of an outbreak. And basically it was, hey, we put our orders in, we just ain't got the stuff. So they had to change platforms. So we've gone through this reorganization of, hey, we can't use that. Let's use this one. Hey, we've run out of that one. We got to quit using that one. Let's use this one. Okay, so that's why we've gone down to the antigen test from PCR testing. It's just because we can get enough supplies of the antigen test to do an antigen test. It's not the best of the platforms we have, but it is a quick way to turn around results and let people know if they have it or not, so they can go home and stay at home for a few days. Okay? So the molecular analysis strand cleavage method uses restriction endonuclease enzymes to cleave the DNA at specific locations. Okay, so we can look at this, and this first picture shows that we have an A and B sample, and they're cleaved at different points. Okay, so we end up separating those sections, right, into a gel, and we can find where they migrate to, and we can see that B had two segments and A had 3 segments. So A got cleaved twice and B only got cleaved once. Okay, so you turn that DNA in with the endonuclease enzyme, you mix it together, and then it looks for certain codes to cut. Okay, so when it recognizes a certain code, it cuts the DNA, and that gives us an idea of what we've got. So used to investigate small genomes, such as those of microorganisms and plasmids. So again, this is in our wheelhouse when we think of microorganisms, used for mutation and polymorphism detection. Okay, because what happens, we have a mutation, that strand changes. Okay, that sequence changes that the enzyme goes in to find to cut. So if the cutting spot has been changed, it won't cut. So that might be like if you look at A versus B, okay, you say, well, maybe there's a mutation between these two, right? Between these two patients. B only gets 2 strands, so there's a mutation where they don't get, where it changes the area that the enzyme normally would cut. All right. So that's kind of the first one. The second one is the hybridization. Okay, so we got this is the second one on 175. And this one, hybridization method, is associated with something called Southern blot analysis. And you may or may not have heard of blot so far. You may have already heard about Western blot. But we're gonna talk about Southern blot first, okay, before we get to Western blot. But Southern blot was first put together not too long ago. Okay, the technique. I want to say, I think I read like in the mid-70s. Okay, so kind of not too far, not too long ago, 50 years ago. So we had Edwin Southern. Wonder why it got called Southern blot now, right? You just thought maybe they just came up with directions, called it Western blot, Southern blot. But actually the guy's name was Southern, so it came up, it was his technique. So if you're good at science, you get things named after you. So this was where we first started doing DNA fingerprinting, okay? So this is how he would do it. The Southern blot analysis starts with restriction enzyme digestion of DNA isolated from a test organism or cell. So we bring in the DNA and we just cut it up. Okay, we do a restriction enzyme to make fragments, and then those fragments are separated by agarose gel electrophoresis. Some of you had been in that. We're going to do a little gel electrophoresis ourselves. But they're separated double-strand fragments are then denatured. So you cut up the DNA and it's still double-stranded, right? You just cut it at certain points. Then you separate the double strand into single strands in the gel. The treated DNA is then transferred or blotted, okay, where it gets its name, from the gel onto a nitrocellulose-based membrane. This is all on 12-9, the look of it. I think that's what we're looking at here. At this stage, all the millions of fragments from the complex genomes will be present in the membrane. So you transferred all the DNA that you collected, okay, is now on this test gel. Look at it that way. All right. So what do we do now? We got all this DNA. We cut it up. It's all present. Then the key to this specificity of the Southern blot technique is the use of a probe. So you hear probe, P-R-O-B-E. So the use of the probe is generally a nucleic acid, several hundred to a few thousand base links, with a known sequence that is used to identify the presence of a complementary DNA RNA sequence of an unknown sample. So what you do is you take if you want to say DNA fingerprinting, and we say we have a crime scene, let's just take the obvious thing here. Crime scene DNA, right? We collected all this body, hair, finger scrapings, whatever, blood, right? We collect all that at the scene and we stored it for years and years and years. And we convicted somebody of that crime. We said, you were there, somebody eyewitnessed you there, and you had a beef against that person. So all that trial came down to a conviction, put this person in jail, and there was no DNA testing. But they collected all of it. And they stored all of it, right? DNA is great because we can keep it forever. Then they bring it in and they say, okay, we got this new technique, Southern blot. We're going to take that DNA, we're going to cut it up, and then we got the person in jail, right? So we get his DNA or her DNA, and we make a probe from their DNA, unique sequence, and then we probe through all that DNA that was collected at the crime scene with the probe of the known person who was in the jail, and nothing matches. We find no similarities. No sequence matches their sequence. It's unique sequence. And what do you do, right? So that's how we came to this DNA fingerprinting, was by doing this, having this probe, breaking apart all the DNA into fragments, smaller fragments, so it could be detected. So we use a unique identifier of the person that we're looking for. And we can do this with microorganisms, right? if we know we have a known sequence of a microorganism and we have a sample from a patient, we want to do this, we would take it apart, digest it, put it all on a gel, and then we would probe for that sequence. So that's kind of how it all kind of got started. We have to get a known sequence, right? So genetic pattern or protein pattern, anything that we've done through all the years is in a database, right? So like E. coli has its own pattern. Staph aureus has its own pattern. So you use that to detect it if it's there in the sample, okay? All right, does that kind of make sense? So kind of where we're headed with all this testing? I hope so. I know it's hard. So the probe and the basis, complementary, the signal can either be by admission of radioactivity, okay? So you're gonna signal on the probe. So if it links something, development of the color, we could do color development. We did that. We've kind of seen that with our ELISA, right? That you just did last week or this week. Remember, we put the horseradish peroxidase, had a primer sitting there on the, and if it was there, if it was still there, then boom, it shows up. Same thing here. Okay. So on 1211, I think we have it. So we have, here's the radioisotope probe here, using it on this side. Here is the conjugated alkaline phosphatase. So that's kind of like where we use horseradish peroxidase over here. And then you had your probe. So the probe will, here's all the DNA sequences in the gel. And then if it matches and stays on there, right, and binds to the complementary strand, then it stays there and we see it in an x-ray and we detect it. We say, okay, we found your DNA at the crime scene, or we found Staph aureus here. Okay, so this is the hybridization method. So this is where this kind of led. This kind of led to this, what's known as a film array or an array method, which allows for multiple targets or samples to be analyzed simultaneously. So if we know, just think of the film array for a blood culture, right? What do you think might be in a blood culture? Are we going to look for viruses? We're going to look for bacteria, right? Yeah, I mean, we could. Blood culture positive. You know, upper respiratory, a sputum culture. What are we looking for there? So all those known probes are sitting in that test kit, sitting in the pouch. And then you put your sample in there and mix it up and wind it up. Okay. Did anything hit? Did anything match? Did anything link? Did anything stay? And if it does, then it's detected, and we'll say, you have C. diff. You know, so that's how these work. So the array method, commonly used for other things, we see for leukemia prognosis, diagnosis of autoimmune conditions, and detection of amplifications or deletions in the DNA, or gene expression with RNA synthesis, or SNPs. And you know what I do is, what is an SNP? Anybody? SNP? Once we get this answered, we'll finish right here. SNP. I want you to answer that for me. Zoom land. All right, so we're going to come back. SNPs, I hadn't heard the answer yet. So deletions in the DNA, gene expression, RNA synthesis, and where there is a single base change. Okay, SNP, single nucleotide base. Okay, all right. Y'all aren't playing along very well on Friday. So we're going to pick up here. We'll pick up here on Monday to show you this is film array analysis. And we're going to show you how this works, and then we'll go into some of the other techniques and work our way through the end of this chapter for Monday and get ready for the test. All right. So we will see you on Monday. We're going to stop the share. We're going to stop the recording.